To begin to introduce this research, first we need to introduce the disease. Huntington's disease is a neurodegenerative disorder where many parts of the brain experience neuron death. However, there is always almost specific cell loss within the basal ganglia structure of the brain. Huntington disease is separated into 4 separate stages of the disease, each becoming progressively worse until eventually symptoms prove fatal, about 20 years after onset of symptoms. The symptoms of Huntington's disease include uncontrolled movements, behavioral changes, and trouble feeding towards the later stages of the disease. The current theorized cause of Huntington's disease is a mutation of the Huntingtin gene, and it is also theorized that the number of CAG trinucleotide repeats present in that mutated gene contributes to this disease.
Our hypothesis states that a FLMHTT mutation causes Huntington's disease phenotype by producing MHTT in astrocytes. So the parts of our hypothesis include A, which is FLMHTT or full-length human mutant HTT. B is the expression of MHTT in astrocytes, and C is the Huntington's disease phenotype. There is a strong correlation found between FLMHTT and expression of MHTT in astrocytes. There is a significant decrease in immunofluorescence BACHD mice compared to wild-type mice when looking at the proteins made, indicating the changes made when FLMHTT is in astrocytes. It was reported that when truncated MHTT is expressed in cultured primary astrocytes in the presence of wild-type neurons, these cells are capable of inducing neuronal death through a variety of mechanisms, including excitotoxicity. This shows the relationship between MHTT expression in astrocytes and how they cause neuronal abnormalities. Finally, there have been a multitude of papers published on the different relationships between abnormalities expressed in the form of Huntington's phenotype and their cellular causes. So for the block mechanism, it is outlined as blocking B should block A, causing C. B being the mutant Huntington protein expression in astrocytes, A being the mutant Huntington's gene, and C being the Huntington's disease phenotype. So to test this hypothesis, we would inject a hormone therapy drug, specifically tamoxifen, to try and stop the expression of FLMHTT in astrocytes, therefore terminating the contribution to Huntington's phenotype. This is hypothesized to block the creation of active astrocytes, which cause neuronal death and other abnormalities at synapses in the Huntington's phenotype. If the hypothesis is supported, astrocytes will terminate expression of FLMHTT that cause abnormalities, specifically in excitatory transmission at corticostriatal synapses. In the paper, this was done, and it was concluded that treating the mice that had the Huntington's gene with tamoxifen had an elimination effect of the astrocytes expression, though not fully, of FLMHTT, thus reducing the overall brain abnormalities of FLMHTT.
For our ideal mimic experiment, we would start by injecting mutant Huntington protein into the brains of 2-month-old wild-type mice. The mice should be allowed to grow and monitored for signs of Huntington's. If our hypothesis is correct, injecting the mutant protein, MHTT, into wild-type mice should cause the HD phenotype to be expressed. The original research worked somewhat in reverse, starting out with mutant mice. They extracted protein from the cortex and striatum of mutant mice rather than adding it, and compared it to normal FLMHT mutations. Through Western blots and probing, it was concluded that elimination of the mutant protein does reduce the overall effect of Huntington's disease.
For the measure experiment, we decided to increase the number of CAG repeats in the mice's HTT gene to theoretically increase the amount of protein secretion in astrocytes. This follows our A activates B template, as A is the mutated Huntington gene, and B is the expression of MHTT protein in astrocytes. We wanted to increase the number of CAG repeats through CRISPR. Once the gene is edited so that the number of CAG repeats is greater than 35, and then the mice have fully developed, proteome-wide quantitative mass spectrometry-based analysis can be performed to measure the secretion of MHTT proteins in astrocytes. Genetically disrupted astrocytes cannot complete their neuron supporting roles, and widespread dysfunction caused by a mutated protein in astrocytes may lead to more rapid or more severe cell death that is seen in more severe cases of Huntington's disease. Typically, the more severe the Huntington's disease, the more CAG repeats will be present. The author of this research paper that we studied is Dr. Michelle Gray. Dr. Gray began her career with an undergraduate degree from Alabama State University, but wrote this paper while at UAB. Currently, she is an assistant professor here at UAB and runs her own research lab. She has recently won awards such as the 2020 Top 100 Inspiring Black Scientists in America, and in the past has won awards such as the 2010 Career Development Award, the Dixon Scholar in Neuroscience from 2008, and the 2002 Ruth L. Kirchenstein National Research Service Award.
The tools outlined in this research paper are not the first time that Dr. Gray has used them. One of the primary subjects the researchers used in this paper was the BACHD mouse model. BACHD stands for Bacteria Artificial Chromosome, which refers to how Huntington gene was put into the mice. This mouse model was created by Dr. Yang at UCLA alongside Dr. Gray working closely on the project. Dr. Gray currently runs a research lab at UAB which specifically studies the pathogenesis of Huntington's disease with a specific interest in astrocytes. She also works on cardiac abnormalities in Huntington's disease, as well as X-linked dystonia Parkinsonism, which is a progressive neurodegenerative disease that primarily affects men of Filipino descent.
In 1993, Michelle Gray began college at Alabama State University in Montgomery. There are several NIH-funded programs Gray was able to get involved in research. She was supported by the Minority Biomedical Research Support Grant and the Minority Access to Research Careers Program. After graduating in 1997, Gray started graduate school at Ohio State University, studying nervous system development in zebrafish. She was the first graduate student in the lab and was funded by the F31 National Institute of Health grant. She explored the supernatory development of motor neurons due to mutations in the deadly 7-Notch1A gene. She found that the extra neurons that form as a result of the mutation are incorporated into the neural circuit critical for escape behavior in zebrafish. This suggests that plasticity or expansion of ancient escape response circuits may have paved the way for larger, more complex escape neural circuits in mammals. She completed her PhD in 2003 and moved to California to complete her postdoc work at UCLA. That was where she changed her study to neurodegenerative diseases. One of Gray's first projects in the lab was working with a team to develop a novel method for sorting and identifying genetically defined cell populations. She applied fluorescence activated cell sorting, FACS, to genetically labeled neurons and then characterized their gene expression profiles, which helped them determine the expression of two different genes necessary for HD pathogenesis. In 2008, Gray published a first author paper in the Journal of Neuroscience, highlighting a novel transgenic mouse model for HD that she developed, a technique used throughout our paper. She studied the expression of the mutant Huntington protein in mice using insertion of a bacterial artificial chromosome expressing the full-length human mutant Huntington gene. It soon became a widely accepted in vivo paradigm to study Huntington's disease pathogenesis and treatment efficacy. She came to the UAB Center for Neurodegeneration and Experimental Therapeutics in 2008. She was promoted to tenure-track assistant professor in 2010. Gray is affiliated with the Center for Gliobiology and Medicine, the Evelyn F. McKnight Brain Institute, and the Comprehensive Neuroscience Center at UAB. She also co-directs the School of Medicine's Summer in Biomedical Sciences undergraduate research program and is on the board of trustees for the Huntington's Disease Society of America. In 2013, shortly after Gray began her lab, she discovered that astrocytes in her mouse model of HD exhibit aberrant glutamate release. Since this is known to injure neurons, this finding pointed to astrocytes playing a potential role in HD pathogenesis. After establishing the role of astrocytes in Huntington's pathogenesis, shown in our research paper, they then explored the potential mechanisms underlying the role of astrocytes. They then went to gliotransmission and inhibited the snare complex, finding an overall decrease in behavioral performance when it was inhibited, though performance did improve by 12 months of age. This suggests that a region-specific approach might highlight the role of specific astrocyte populations in the pathogenesis of Huntington's. Dr. Gray is currently continuing to study the contribution of astrocytes to Huntington's disease pathogenesis. Quote, "We believe by focusing on one cell at a time, we will be able to identify novel mechanisms to target for disease intervention."